I just have to say, wow. What a wonderful, stimulating way to start our day. Thank you for that great presentation. And also thank you for your leadership for HEROES too. We really appreciate that. My name is Iain Cheeseman. I'm a professor in the biology department and at the White Institute, and I'm also one of the associate directors for MIT HEALS. And I have the privilege of being able to introduce you to our session on seed grants and showing you some of the things that we've funded thus far. And I think as you've heard today already, I think that concept of we're not going to solve the most challenging and tough scientific questions in health and life sciences if we just keep doing the same things in the same way. And so from my perspective, certainly for my own work is focused on how human cells function, how they grow, divide, and do the things they need to do. And particularly, we're looking to find those hidden proteins that Nubar mentioned, the sort of hidden elements that drive that forward so we can understand rare disease and cancer and other components. I think that the most important things for our work recently have come from that convergence that Angela mentioned at the beginning, that ability to bring together different ways of thinking about a problem and approaching that problem. And so I think HEALS is really looking to sort of empower and enable that ability to bring things together in new ways. I think that there's two fundamental challenges for this, though. And the first is, as you heard from Sally this morning, 75% of MIT's 1,000 faculty are working in these areas. But how do you even begin with that? I know the biology department really well, but actually the landscape of all of that other work at MIT has been much harder for me to unpack. And actually seeing that potential over the past year, what HEALS can do and enable has really blown my mind. And so we're really looking for new strategies to be able to think about how to cross those traditional boundaries, whether those boundaries be fields or whether those boundaries be departments, and bring people together in new ways, crossing these disciplines in different ways. And I think actually for my work, someone we've been working with for the past few years now, like the biggest thing that I suffer every day is imagining, what if we had just met each other 2-3 years earlier than that? How can we find those new connections between disparate areas? I think there's not only a challenge in being able to find those connections. What is the most impactful thing for the work that you're doing, and how do we combine that work in new ways? There's also big challenges for funding that. And I think that we've heard already again today too, there's major limitations even in the, a year and a half ago, how some of the conservative systems work for how we fund that work. And so we really want to be able to solve both of these, and the seed grants are one of the ways that HEALS is trying to achieve that. Can we encourage MIT faculty and PIs to come together in new ways? Can we also provide that initial support that's needed for that? So the MIT HEALS seed grants were designed for this purpose. We're really seeking to make MIT faculty make new connections across departments and empower them to take bold new directions for the work that they're doing. We currently have two different types of grants for this. There are breakthrough grants that are composed of teams of 4 to 5 MIT faculty, have about 2-year attempt to be able to go this. And we have smaller innovator grants, and these are teams of 2 to 3 faculty and PIs who are looking to sort of test out and combine new areas. Over the first year, we got more than 80 applications across these different funding schemes. And we were really fortunate with the great support that MIT has been able to initiate for this to be able to fund 17 of these total projects. This is 2 of these breakthrough awards and 17 of these innovator grants. In total, we've been able to support through this mechanism 48 different faculty across MIT. And so this is already having a very large impact on touching a number of different departments, more than 12 different departments already represented with these funding streams. So of these 19 overall funded proposals, we're going to hear about 5 of these today, and I'm really excited for these too, just to be able to hear these presentations. These projects cover a really exceptional range of science and engineering and different approaches and different disciplines, different questions, some much more connected to really making that immediate tangible impact, some still in that very initial discovery phase. And so I think that this selection of talks you're about to hear will be a really good summary of where we're touching on that. So we're going to begin. We start with a wonderful presentation from Polina Golland, who is leading one of the Breakthrough Award teams. Thank you very much. Looking for your time.
Good morning, everybody. So we are one of the Breakthrough Awards. I should say that I knew all of the collaborators before, but never worked with them before. So HEALS basically created something that didn't exist before. The goal of our project is to map out dynamics, to enable mapping out of the dynamics of pregnancy processes. So understand better how maternal and fetal systems interact with each other. And the key idea is to enable continuous mapping by non-invasive sensors that might be low information by themselves, but combined with AI and high information sensors when we train the AI systems can help us reveal important properties about the processes. So I have this fun task of telling you what 5 disjoint groups up till now came together to do in 7 minutes. And so instead of telling you 1,000 words, I want to start with this picture. So this is the 5 PIs, Tal Cohen, Shana Zhao, and Polina Golland, and Alan Yasinov are all building new sensors that will enable us to measure things non-invasively about the processes that are going inside the maternal body, the maternal brain, and the fetal system. Tal, myself, and Alan build dynamical models based on biomechanical insight and AI approaches to take that data and to take the data from the sensors and actually tell us something about these processes. And Alan and I have been doing a lot of MRI imaging in animal models and in pregnant women to get very high spatial and temporal resolution data that would be completely implausible to get over long periods of time. You can't put a pregnant mother in a scanner for a week. Half an hour is a stretch. But by learning from that MRI data combined with AI, we can transfer some of the signatures that we're looking for to the non-invasive sensors to actually enable continuous monitoring in both animal models and pregnant humans. So the long term here, as I already told you, is to build sensors, and we believe that these will dramatically change how we image pregnancy by, again, uplifting non-invasive measurements to high precision models using MRI and other high information invasive measurements in animal models and AI approaches to merge what we image in real time or what we measure in real time. To me, every measurement is an image, sorry, right? So to what we measure in real time in the pregnant organisms. Now, we're going after 2 long-term goals. The first goal is to get biological insights into how pregnancy evolves, how brain and uterus interact, how processes initiated in the uterus through placenta affect the fetus, and moreover, understand sort of the entire system. It's amazing how little is known about pregnancy. Quite often, when we do our research and find new insight, we go back to biology and medical literature and discover that, well, we can't actually say if this is in agreement with previous knowledge or contradicts it because we don't know. And our second long term is to use these devices to support pregnancy, to support high risk pregnancy, to understand when it is that we should deliver a baby, or rather our collaborators should deliver a baby versus leave it in. Right now, quite a lot of it is based on empirical intuition, which is great, but how do we quantify that process and provide better insights into the process of nurturing and delivering healthy babies is another important goal. Okay, so that sounds very lofty. What have we done this year? We seeded 5 projects that sort of spontaneously happened over the summer. The ideas for these projects came up because we came together and spent hours brainstorming about what could happen. So each one of these projects has at least 2 groups from our strange, chaotic group. And each project brings together either two novel imaging modalities or a novel imaging modality and an AI approach to support it to provide important evidence. We are already up and running in terms of making measurements in animal models, and we're working on the IRB to actually do measurements in women non-invasively at Boston Children's Hospital. We've also connected to Boston Children's Hospital on running some experiments in sheep, which are the closest model to human pregnancy. So we're sort of spanning everything from mice to sheep to humans, and very non-invasive surface-based light touch measurements that can be done continuously all the way to MRI with biomechanical models and AI sneaking in. So I hope to tell you more about these projects next year, but I want to leave you with a picture of our team. So this group didn't exist until we got the grant and started cranking on it. It includes 5 PIs. Officially, MIT HEALS funds us at the level of about 1 graduate student or a postdoc, but it turns out that every group had enough interest that we are now somewhere between 15 and 20 people who come together every month, consume large amounts of pizza. The meeting is an hour and a half. The PIs are invited to the first half hour, and then we leave. And then real stuff happens, right? So it's been amazing to see students connecting from different projects and starting to cook up projects and ideas that we didn't even think about in July when we conceived of this project. So I'll leave you with this group that is now together working for the next 2 years to bring new measurement mechanisms to mapping pregnancy processes and ultimately affect human health. Thank you.
Hi, good morning, everyone. Thank you for the invitation to speak here. I'm delighted to present some of our work on a HEALS seed grant between myself and my colleague, Professor Sangeeta Bhatia. And we're working on elucidating links between hemodynamics and Fontan-associated liver disease. So the clinical problem we're working on, it's a congenital heart defect where kids are born with one functioning ventricle and the other one isn't functioning as well. So kind of half a heart that works. So the pumped systemic is great, but the pump to the lungs is not working. There's about 1,000 surgeries called the Fontan surgery. This is the last of 3 surgeries to reconstruct their vasculature. About 1,000 per year in the US. And there's about 70,000 of these people, of these patients living at the moment. And it's expected to double in the next 20 years. These surgeries are getting better and the patients are surviving longer. This is really interesting from a mechanics perspective because instead of 2 pumps in parallel, you have this 1 pump and no pump to the lungs for oxygenation of blood. It's also heavily influenced by breathing. So when these patients inspire, the thoracic pressure drops, and this causes a forward flow of blood to the lungs. But when they expire, the opposite happens, and they actually get this reverse or retrograde flow to the abdominal organs, and this can be problematic in the liver. So instead of their right-sided heart function and their flow being synchronized with heartbeat, it's actually synchronized with breathing. So very unique hemodynamics and blood flow patterns. And we're interested in how those blood flow patterns lead to the comorbidities that these patients live with. So one of those potential implications is something called Fontan-associated liver disease. And this is where these patients have liver fibrosis, similar to alcoholic liver disease or different types of liver disease. And there's a knowledge gap there relating to how the blood flow patterns or the hemodynamics contribute to that type of liver disease. And that's what we're trying to address with this work that we're embarking on. Is it the elevated pressure in the abdominal organs, or is it the reverse flow that happens with every breath that contributes to this liver disease? So our lab is working on some robotic models to mimic this mechanical physiology, especially the interactions between the cardiovascular and the respiratory system. We've built a robotic system that can mimic diaphragmatic function, mimic blood flow, and mimic ventricular contraction of this single ventricle disease. So this helps us to elucidate insights on the bench. But of course, we want to validate them clinically. And we've had the privilege of working with a group of patients at Boston Children's Hospital to perform MRI studies, more MRI imaging. So we have patients that have had this surgery go into an MRI scanner and perform different breathing exercises, and also we have them exercise within the scanner, and we can measure the flow in their vasculature. So this MRI study, we have an AV guide so that they breathe in a specific way, similar to what you might have on your Apple Watch for breathing. And we have a visual guide for exercising, and we've built an MRI-compatible ergometer that can go fully in the scanner. And then we measure flow in their IVC, which is the surgically reconstructed shunt that these patients have. We've had some interesting findings relating to the interaction of breathing mechanics and flow in these patients. As you increase respiratory effort, so breathe deeper, the reverse flow to the abdominal organs increases, and that's at a fixed respiratory rate. Alternatively, when you keep your effort constant but increase your respiratory rate, you also see an increased reverse flow to the liver. So these are interesting findings that were kind of new to our surgical collaborators and are something that potentially we can intervene on, both non-invasively and with devices. We also had these patients exercise within the scanner, and we showed that with increased exercise, increased cadence, or increased resistance, you actually reduce that reverse flow. So this could be another interesting intervention. We also look at ultrasound because MRI imaging is cumbersome and costly, and the patient is supine. So we're looking at using ultrasound and really honing in on the hepatic vein. So what's the flow to the liver here? With ultrasound, we can really show this color change indicates a change in direction of the flow. So we really show again that this flow happens with each breathing cycle. There's flow going to the liver that shouldn't be. And we can quantify that pretty nicely in these patients. And this enables us to do this imaging in different circumstances when they're standing or exercising, and really understand the parameters that are associated with this. Now we want to use these findings to really understand the gap of why these hemodynamics are causing liver disease. And that's what the focus of this MIT HEAL Seed Grant is. So it's a real pleasure to work with Professor Sangeeta Bhatia and her group, especially postdoc Vardman, who has been helping us with this. The goal is that we take our curated clinical data that we're working on with our Boston Children's collaborators. We have computational models that are lump parameter models for systemic flow. And we work with the Bhatia group to develop liver-on-chip models where we can use our flow data from the clinic and impose that on a liver-on-chip model and really decouple, is it the high pressure in the liver? Is it the reverse flow to the liver? What is causing the disease? So this really is a way to bridge that gap in the knowledge and use imaging, both ultrasound and MRI, to understand the mechanisms of Fontan-associated liver disease. It's been a pleasure to learn the processes involved in creating these organs on chip, these liver spheroids that Sangeeta and her group are expert at, and to figure out how to perfuse them with flow so that we can answer these questions. My group, postdoc and Andy and a grad student, Eve, have been working with her group to get these liver-on-chip models up and running, and we're really excited to do so. And in the end, what we're excited to do is to relate hemodynamic imaging data to end organ function. And the ultimate goal is that we can then intervene. So if we know that this reverse flow is leading to liver disease, can we build devices that go on the surgically implanted shunt and prevent that reverse flow? And they're synchronized with breathing, harvest energy of breathing, and prevent the comorbidities that exist in this patient population. So thank you for listening, and thank you for the support of the seed grant.
I want to first thank the organizers for the opportunity. I'm very excited to be here to share our collaborative work with Professor Ed Boyden in developing an in situ single molecule protein sequencing platform. And so before going into protein sequencing, I just want to first describe in general what our group is interested in as a context. So we're very interested in building new tools, both chemical and physical tools, so that we can better understand protein dynamics. And many people in this audience are probably familiar with this concept of biological structure and function relationship. That is, to understand the function of a protein, you want to first get the atomic structure of this protein. But I will point out that it's not always obvious, right? If you look at the structure of microtubule binding, kinesin microtubule binding domain, it's not very clear how it can actually carry out its function, which is to carry cargo transport. But instead, if you have this kind of movie that shows you directly the motions of these proteins, then it becomes easier to understand. And that's why we're very interested in developing tools so we can directly visualize individual molecules. By taking movies of these molecules, we can learn molecular mechanisms by which these proteins function and how they cause human diseases when dysregulated. And specifically, we're looking at single molecule fluorescence microscopy that can allow you to directly look at individual molecules. And this technique has really answered a lot of important biological questions in the past 2 decades. But there is still one significant limitation, and that has to do with the stability of the imaging contrast we're currently using. So here I'm showing you a movie of single individual organic Cy3 dyes. Each one of that bright spot corresponds to a single molecule Cy3 dye. And you can see that in this movie, as time goes on, their signal disappear within just a time scale of a few seconds, which means that it's very hard to actually capture the entire biological process. That's why I'm going to introduce you to this new probe that we're developing in our group that is a nanoparticle doped with rare earth and lanthanide ions. And as you can see in this movie, they're extremely photostable on the time scale of minutes to hours, to days, to even years. And I promise you that I'm playing a movie, not just a single snapshot. And so with this kind of much improved photostable probes, we've been able to study a particular protein, membrane protein called EGFR, which is very important in cancer signaling. And so you are seeing in this movie, each individual bright spot corresponds to a single EGFR, and they're quite dynamic in the membrane of a live cell. This is a movie over the course of 15 minutes, and you can really see very clearly the motions of these proteins to understand protein-protein interactions and how they respond to different inhibitors, for example. Now, of course, many biological processes involve not just a single type of protein, but many different types of protein, they all come together. So in order to fully understand these processes, we want to be able to map out multiple different types of protein as well as their locations. And the locations are important for their function, right? For example, this MMDA receptor, when they signal within the synapse, they can lead to neuronal health. However, if they signal outside of the synapse, they can instead lead to neuronal death. And so it's very important to be able to simultaneously map out the protein sequence so we can get the identity as well as their subcellular localization. And currently, there's no single technique that actually can do this at the single molecule level. For example, for mass spectrometry, we want to be able to increase the sensitivity down to single molecules. For immunostaining, we're currently using multiple rounds of imaging and staining, and also we have to rely on very robust antibodies. And that's why in this HEALS project, we're working to develop a technique that can allow us to do single protein sequencing. And this was inspired by a technique called fluorosequencing done by Eric Ansland, UT Austin. And so in this method, they label every cysteine residue within a peptide with an organic dye. And they use a chemistry called Edmond degradation that can sequentially cleave off the N-terminal amino acid. And so by the time that a cysteine residue is cleaved, you will see a signal loss in the fluorescence time trace. And by using this kind of information, you could, in principle, reconstruct the protein sequencing. However, in this work, they also identified that most of the dyes, except maybe exception of 1 or 2, can survive this really harsh Edmond degradation chemistry. So most of the dyes will photobleach in this process. Also, if you're using dyes, there are only a handful of dyes that you can simultaneously image, maybe down to just 3 or 4. But there are 20 different amino acids that one has to image in order to reconstruct the protein sequencing. And so in the interest of time, I won't go into the technical details, but I'll just show you that we can now simultaneously image 20 different colors at the same time down to the single molecule level, and we can watch their interactions in living cells. And so by combining this kind of 20-color imaging technique with Edmond degradation, we can now, in principle, for example, you can label every one of these 20 amino acids, at least the reactive ones. And then you can do sequential element degradation. And by watching the signal decrease in different color channels, one can in principle reconstruct the protein sequencing. Now, of course, since we're using optical microscope, our spatial resolution is limited by the so-called diffraction limit, which is on the order of hundreds of nanometers. That means if you have 1 molecule or 10 molecules all within this diffraction limit, they all appear as this blurry bright spot. And so in order to resolve individual proteins inside cells, we really need about 1 nanometer spatial resolution. And this is where Ed comes in. So Professor Ed Boyden has already pioneered this really clever form of super resolution microscopy called expansion microscopy, which literally physically expands the sample, your cell. And so now you can directly watch individual molecules with much greater clarity. And they have been able to show that you can see subcellular structures. You can even get structures that are really close to cryo-EM structures. Recently, they've also developed a new form of element degradation chemistry that is compatible with expansion microscopy. And so we're integrating techniques from different tool labs to build this in situ protein sequencing platform. We're going to anchor our proteins in cells. We'll expand them in this expansion gel. We'll label with amino acid specific probes with different colors. And then we do sequential element degradation to read out the sequences of these proteins. And so I'll just finish by saying that this is really a high-risk, high-reward project that requires expertise in many different research fields in chemistry, physics, biology, and computation. And so we're extremely grateful for the support from MIT HEALS that allows us to bring together these many different talents. So shown here, William is a graduate student in my lab, primarily working on this project. Joao is also another graduate student supported by HEALS, and of course, our collaborator, Ed, Camille, and also Sarah. And thank you for your attention.
Hello, good morning. I'm happy to be here today to introduce our project, which is sort of an unusual project for HEALS. I think we're representing the life sciences part of the acronym. Because it's a collaboration between the Van der Heyden lab and my lab. And if you look at, these are our opening websites, and you can see that we come from very different fields. Matt is a cancer researcher, and I'm an oceanographer. But we're united in our interest in metabolism. All of life has metabolism. The only difference is we're interested in the metabolism of the oceans and not, well, we're interested in the metabolism of humans, but our work is the metabolism of the oceans. So we have the questions about what's going on in the oceans, and Matt's lab has the techniques to help us answer those questions, metabolomics. First, let me just introduce that the ocean ecosystem, when I look at seawater, I really think of information. There are millions of unique genes in the microbes in the oceans. And the phytoplankton are the base of the food chain there, and they do half the photosynthesis on Earth. We call them the invisible forest. And they're generating this incredible diversity of organic compounds that feeds all of the other microbes. And there's 700 petagrams, that's 10 to the 15th grams, of dissolved organic carbon in seawater that comes from this metametabolism. So how do you begin to figure out what's going on? Well, we start with a simple model system, which is Prochlorococcus, which has been my muse for the last 40 years. It's the smallest and most abundant photosynthetic cell on the planet. It's less than a micron in diameter, and it's the same size as the wavelengths of light that it absorbs. It's an amazing little photosynthetic machine with 1,700 genes. It can convert CO2 and sunlight into life, which is something we can't do yet. And there's hundreds of different genomic variants. Their collective genome, ocean-wide, is about 80,000 genes because there's such incredible diversity. So we started, when we first were able to isolate Prochlorococcus from seawater, we transfer them regularly in liquid culture. They won't grow on plates, so you have to keep them going. You have to keep transferring them. And they bring along with them the bacteria that depend on their organic carbon, which you can see here. The rod-shaped ones are the contaminants, and the round ones are Prochlorococcus. And so there's no organic carbon in the media, so Prochlorococcus is feeding these cells. And it's what we've learned recently, going back into the archives of our frozen samples, that they curate this community of microbes that they keep with them as we transfer the cultures over many, many, many years and many generations. This is making me feel old. But anyway, I've been here a long time. So we're really interested in what the exchange of metabolic products is between Prochlorococcus and this community of what we call heterotrophs that depend on them. So what is this collective metabolism? And that's really at the heart of our project. And up until recently, until this collaboration, we depended on looking at transcriptomes to understand how these were talking to each other. In fact, we published a paper called Torn Apart and Reunited, where we took them apart and we looked at their transcriptomes and put them together. And that gave us hints about how the heterotrophs are increasing the fitness of Prochlorococcus. We know Prochlorococcus is feeding them dissolved organic carbon, but what molecules? And we think, have good evidence that there's a return of those molecules back to Prochlorococcus. In fact, if you grow Prochlorococcus and then you put it in extended darkness, it will die unless it has its heterotrophs with it. And we have good evidence that suggests that the heterotrophs actually are feeding Prochlorococcus during the dark. I think of them as a pantry. Prochlorococcus gives them its organic carbon when it photosynthesizes during the day, and then it can recoup some of that carbon at nighttime, or when they're stuck in extended darkness, for example, if they get mixed down into the deep ocean. So this is our HEALS project, which is what are the actual metabolic exchanges, moving from transcriptomes to actually looking at those molecules. So what is Prochlorococcus giving the heterotrophs, and what are the heterotrophs giving them back, and how is it influenced during darkness? Now, the challenge, that sounds like a really simple, straightforward problem, especially compared to listening to the talks this morning. But the challenge is that Prochlorococcus is not E. coli. It grows really slowly. Its growth rate is 100 times slower than E. coli, and its dry mass per milliliter is an order of magnitude less. So it's very hard to get large concentrations. Also, we have this seawater in the way, which is not good, I learned, for the mass spec. So long story short, through much trial and error, we've been able to develop a protocol. Christine Ziegler and Mohammed in Matt's lab developed this protocol, and the short version from this slide is that it works. We can now measure metabolites, and we have some preliminary data that is identifying amino acids that are flowing from Prochlorococcus. The heterotroph is putting out some nucleotides, and there's something going on with the B vitamins between the two. So I'll end with just this overview slide, which is my fantasy world, where the life sciences borders between these different levels of organization in the life sciences start to dissolve a little bit more, because we've learned a lot down here at the reductionist end of the life scientists. That is how life works, which this project is just amazing in unveiling. But life is a complex, self-organizing system, the biosphere, and everything that emerges at these higher levels of organization, those properties feed back and design the lower levels of organization. So I think of it in this population and community dynamics is really the why things work the way they work. So we need to, I learned studying Prochlorococcus that if I have it in pure culture versus if I have it with one other microbe, I get a completely different phenotype. So Context is all. Thank you.
Great. Okay. So it's been a long and amazingly inspiring morning. And for the last talk of the session, we're going to play a bunch of games. Okay, because this proposal is using social interaction games, and I'm going to teach you about them by letting you play them. Okay, so we're going to try out this game. All of you are going to make a series of choices, playing the game against me. I'm going to be your opponent in this game, or partner, depending on how you think about it. Okay, on each trial of this game, you're going to get a choice between a split of a pot. There's $12 available on this trial. You're going to put up your hand when I ask you. Everybody has to play. You are going to choose either part A in the gold or part B in the silver. And I'm going to also choose secretly part A or part B. And here's how the payoff works. If we choose opposite parts, we both get it. If we choose the same part, no matter which part, we don't get it. Okay, so you have to choose opposite to me. Everybody ready? Okay, I have secretly made my choice on trial 1. I've chosen either A or B. Put up your hand if you've chosen A. Put up your hand if you've chosen B. Okay, I choose A, so you made money if you chose B. We're going to play again. Put up your hand if you've chosen A for $12. Put up your hand if you've chosen B for $8. Okay, I chose A, so if you chose B, you made money. We're going to play again. Ready? Put up your hand if you've chosen A for $2. Put up your hand if you've chosen B for $10. Okay, I chose A, so if you chose B, you made money. Oh, no, sorry, I chose B on this one. I chose B. If you chose A on this one, you made money. Okay, last trial. Keep track of how many times you've won. This is the last one. Ready? Okay, there's $10 and $20. I've made my choice. Put up your hand if you've chosen A for $10. Put up your hand if you've chosen B for $20. Okay, I chose B, so if you chose A, you made money. Okay, who made money on 2 trials? 3 trials, 4 trials. Okay, great. That's the game that we're using to study how anxiety influences social interaction. Now, why does this matter? Well, first of all, I just want to say this game is hard. So when we play this game, our participants play against 4 different partners interleaved. So you don't always play against me. It was a little easier playing against me. You got to kind of know my personality. But if you were playing in our game, you'd be playing against 4 different partners interleaved. One partner is like me, always takes the big pot. 2 of the partners change. They're very hard to predict. And one of the partners always chooses the small pot. So you can learn that you can always get the big pot playing against them. The one who always chooses the small pot is the easiest to play against. The dynamic players who change are hard to play against because they're unpredictable. But actually, the player like me, the one who always chooses the big pot, is also really hard to play against. And so on average, people in our game make money on just over half of trials. So those of you who made money on 2 trials, that's about average performance. But the thing I think is most interesting about this plot, so each dot in this plot is one person who played our game. And you can see that in particular, when playing against partners who always take the big pot, our participants were very variable. Right? Some people learned quickly and made money on every single trial. That's like those of you who made money on 4 trials. But some people, despite playing against this partner who is 100% predictable, always chooses the big pot, some people never made money at all. 40 trials in a row, they were like, nope, 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 I am not letting you get that big pot. Okay, so it's that variability in how you play against these different partners that we're using to leverage understanding the underlying mechanisms that people bring to bear on games like this. Okay, so part of why this is interesting is because if we ask people in standard clinical measures to self-report their own levels of social and general anxiety using standard survey measures, levels of social anxiety and generalized anxiety are correlated with each other, very correlated. People who report high levels of one report high levels of the other. You may also know that anxiety rates are going up really fast and in ways that are hard to understand. So anxiety, particularly among teenagers and young people, is exceptionally high right now. So understanding the underlying mechanism of anxiety is related to a very large mental health burden. But despite this high correlation between social anxiety and generalized anxiety, we hypothesize that they reflect different underlying mechanisms in the mind and brain, and that sorting out those mechanisms will be important for improving treatment. Okay, so how do we test that hypothesis? Well, that's where we're using play in our game, and in particular, a computational model of the calculations that your brain had to do in order to figure out how to play against me. I'll just briefly show you that in our behavioral data, so this is just from about 1,000 people who played the game like you, but on the internet, you can see that self-reported social anxiety is correlated with making money on more trials, whereas generalized anxiety is particularly with the low bidding partner, correlated with making money on fewer trials. So we can start to disentangle the effects of anxiety in this game. I'm not showing the computational model that explains this, but trust me, we have one. And then the amazing thing that HEALS is letting us do is take this game into an MRI machine so that we can begin to disentangle the separate neural systems that contribute to each of these components of play. In terms of where we've got to, so we spent the fall setting this project up, and as of last week, the first participants were in the scanner over at the Martinez Center. So thank you very much for that. The team doing this project is bringing together 3 separate areas of expertise necessary for this project. Rahul Bui, who's in the Sloan School of Management, is an expert in behavioral economics. Amrita Lamba is doing the main work. She's a computational psychiatrist. And then there's me. Okay, and with that, thank you very much for your support.